Hey, API biology. All right, let's talk about the rest of some more of the notes. I want to talk about nucleic acids right now. Nucleic acids are, I mean, they're the coolest things ever. I love nucleic acids. It's just got all the chemistry and it's the heart of all biology, and they're just so great. Look at how beautiful they are just looking. Okay, nucleic acids are primarily used for information storage and transfer. You also get ATP, which is for energy transfer, which we'll talk about later. There is also, yeah, there's just different kinds, and they're all super great. Nucleotides include a lot of different biological macromolecules kind of combined together, or the monomers. So every nucleotide has three parts. It has the phosphate group. Phosphate, this is a chemistry thing. Whenever I see 8 at the end of something, I think oxygen. So phosphate means phosphorus with oxygen. Nitrate means nitrogen with oxygen. Sulfate is sulfur with oxygen. So that's the phosphate group. It is then linked to a pentose sugar. So this is a carbohydrate. It is a saccharide. Specifically in DNA and RNA, it's either ribose or deoxyribose, which I'll show you the difference in a second. And then there's also a nitrogenous base. It's nitrogenous because it has a lot of nitrogens. It is called a base because it is a base unit, but also because it has a pH that is, like, if these things are just floating around in water, they will make the solution that they're in more basic, less acidic. So it goes both ways. So those are the three parts. How they differ and how they stay the same is what makes all the difference. So the phosphate group is always identical in all nucleotides, in all nucleic acids. Nucleic acid and nucleotide are synonyms to each other. So if you hear either one, it's the same thing. The nitrogenous base is definitely different. The sugar is very similar. There's some slight differences between RNA and DNA, but it's pretty much very, very similar. So this is the three parts. When they bond together, they become DNA or RNA, the polymer sort of versions of those things. You can call them polynucleotides, but no one really does. I guess I've never really heard it used, but theoretically that's what you could call them if you have a whole bunch together. So here's the polymer structure. All right, and this is a great example because you can see a lot of the differences. So when they polymerize, then they become a polymer. The bond is always formed here. And there's another diagram later on that shows this really well. So let me actually switch down to that one real quick. Excuse me. Okay, it's always formed at the very bottom of one of these ribose or deoxyriboses with the phosphate of the next one in line. So this directionality is actually really important. We call it a 5' and 3' end. Those come from the sugar. So the sugar in RNA is called ribose. So this is a ribose molecule. RNA stands for ribonucleic acid, meaning ribose, like nucleic acid. So it has a nucleotide, it has a ribonucleic acid. DNA stands for deoxyribonucleic acid, which means ribose, but missing an oxygen, deoxy. So if you compare ribose, you'll see that there's an oxygen right there. And if you compare that exact same deoxyribose in DNA, there is no oxygen right there. That is one of the key differences between RNA and DNA. What really this means is RNA is less stable and DNA is more stable. And that is one of the key reasons why. This extra oxygen is a pretty big atom compared to just this hydrogen by itself. So it kind of makes those linkages, it makes them kind of wiggle around a little bit more, and it's harder for them to stay in a nice straight line. At least that's how I usually think about it. So DNA is more stable than RNA, and there's a reason for it. It's because of that lack of oxygen. Really helps when you have all these molecules close together. Anything that's big that can get in the way is going to make it less stable. We call it steric inhibition, or it's like a sterically inhibited, but that's a whole nother chemistry word. That's organic chemistry, fun stuff, but yeah. Okay, so there are different bases. So you can see here, adenine and guanine are both pretty big. They have a 5 and a 6-membered ring fused together for both of them. They are structurally different. You do not need to know the difference. That is not important. If you don't have to memorize these or anything, because that would be unfortunate. Cytosine and thiamine are just the 6-membered rings without the 5. And this is how, when you get down here, they link together. A big and a small link with a big and a small. So it's A and T always link together, and C and G always link together. Another difference between DNA and RNA is that RNA does not have thiamine. It has uracil instead. And you'll notice there's only one tiny difference, and it's that right there. There's a carbon there. It's a methyl group, a CH3 group. Here it's just a hydrogen. So it's a very tiny, tiny, tiny difference. Oh, I should mention, when you're looking at diagrams like this, anytime you see an intersection, like here or here or here, there's a carbon there. We just assume the carbon is there, but we don't like to draw it. This comes up in organic chemistry and biochemistry a lot, where there's just so many carbons. If we wrote every single one, it would look like an absolute mess. So we don't write carbons unless they're off, like side chains like this. And in these little corners, you would write a nitrogen to show that it's a nitrogen and not a carbon. So ribose has 1, 2, 3, 4, 5, 6 carbons. And those are those all in a row right there. 1, 2, 3, 4. Oh, it's 5. I'm wrong. Sorry. 1, 2, 3, 4, 5 carbons. That makes more sense. 6 didn't make sense. So ribose has 5 carbons. 4 of them are part of this pentose, pentagon-shaped ring with the oxygen up there. One of them is kind of off in a side chain off by itself. Okay, so let's talk about this 5' 3' end thing. So it actually is super important. Okay, so when these link together, they always link with the 3' carbon, which would be like over here. The oxygen that's connected to the 3' carbon is connecting to the phosphate of the next nucleotide. So we call it 5' and 3' because in biochemistry and organic chemistry, when you have many carbons in rings like this, there's a certain way that you label them. So like this is the first carbon, second, third, fourth, and fifth. So it's saying which carbon are we seeing? So there's always one strand that runs 5' to 3'. So there's like the free 5' up here and the unbonded 3' down here. And then the connecting string, the big thing about DNA, which I think you know this already, is that there are 2 individual molecules that connect with each other. And we say that they run anti-parallel to each other because they run in the opposite directions. So 5' to 3', opposite 3' to 5', and they smack together and connect with each other. All right. Each strand of DNA is a completely separate molecule. They have covalent bonds. Oh, these covalent bonds that connects them are called phosphodiester bonds. I don't think it's going to come up, or phosphodiester linkages. Just in case that's important to you, that's what they're called. An ester is a type of bond. It's a bond with oxygen in the middle. Again, it's like organic chemistry terms here. And it involves a phosphorus, so it's a phosphodiester bond. It's a whole thing, just in case you're curious. So that's the thing, that's what keeps this this phosphate background or phosphate ribose, phosphate sugar background, because it's got phosphates, sugar, phosphate, sugar, phosphate, sugar. So this is the backbone. It's the same on both sides. The only real difference, again, is these bases. So the bases are connected. They are not bonded together. They are attracted to each other by hydrogen bonds, which is why they've got these little dashes. So the same hydrogen bonds that keep water together are exactly what keep DNA together. So they're not really bonded to each other, but because there's so many linkages, it's insanely, incredibly strong. It is so difficult to tear two DNA strands apart from each other. You can do it chemically with enzymes, which is how the two strands are split apart to replicate DNA or to translate or transcribe it into RNA. You can also do it with heat. So if you have, especially a smaller section of DNA, you can, we call it melting. You just raise the temperature and the two strands will separate. The backbone won't. The backbone you have to do it's much harder to do that. It's definitely possible. There are ligases and a whole bunch of enzymes that'll snip the backbone all over the place, but it's a covalent bond, it's really tough. Okay, another thing you're gonna notice is that A and T, adenine and thymine, have only 2 hydrogen bonds that connect them, but guanine and cytosine have 3. So that definitely has an implication, something to think about, what that might mean. Okay, so I just said they're 3' to 5', 5' to 3'. They're anti-parallel. They're not, perfectly lined up. They're opposite. It creates this a double helix. It's actually a specific type of double helix. I'm going to go back to the book because I can't remember what it's called. It has a specific name. It's not a perfect double helix. You might not need to know it. I think it's like an alpha helix or a beta helix. There's some word for it. But anyways, you'll kind of notice it's not like a perfect spiral. Like you can see that it's like a ribbon that's been twisted. It's a little bit closer here and there's a wider gap there. Again, this is all based on, is determined by all of the molecules, the phosphates, and how they all line up with each other. It's really useful because other molecules or macromolecules or proteins or histones or all sorts of stuff can attach to DNA based on the exposure of those different base pairs. Yeah, so we call those base pairs. You guys should all know ATGC. Absolutely something you have to know. Adenine, thymine, guanine, cytosine, uracil. So that would be AU. I always remember it because G and C both kind of have they're like curved shaped letters. So it's like G and C click together. And then AT, I don't know. Tata boxes remind me of it. I guess AT-ATs from Star Trek or Star Wars. Ooh, not Trek. Ooh. I'm gonna piss someone off there, mostly myself probably. Okay, I'm trying to think if I know anything else about DNA. It's awesome. It's the best thing ever. It's super cool. Okay, DNA versus RNA, also super important to know. DNA tends to be in that double helix. This helix, you can tell, is not drawn as accurately. This is a much better representation. You can kind of tell there's some subtle differences, but it doesn't matter. It's fine. RNA tends to be single-stranded. You actually can have double-stranded DNA, but it's not very common. Again, the extra oxygen in the ribose gets in the way. It makes it harder for that to happen, but it's possible. They both all have cytosine, guanine, and adenine. RNA has uracil instead of thymine in DNA, and the sugars are different. So that's kind of a nice little summary there. All right. I think that's all. Let me just double check to make sure I don't have anything else super exciting to say about DNA, nucleotides. Oh, this is kind of fun. G and the smaller ones, C and T, cytosine and thymine, are called pyrimidines because they only have one 6-membered ring. The sugar on them is the nitrogen base is a 6-membered ring of carbon and nitrogen atom. The purines are adenine and guanine. I don't know if you need to know the words purine and pyrimidine, but there they are just in case they seem to mean anything to you. So C and T are pyrimidines, and A and G are purines. I do remember that. That is definitely interesting. Okay, well, let's stop the notes right there and let me show you a couple of quick things. I'm going to go to my YouTube channel again to just show you videos that might be of interest. Excuse me, I got to pull it up. Okay, so my YouTube channel, here it is. There is a video from AP Biology that's all about proteins and nucleic acids, so definitely worth a listen, a watch. Again, the first probably close to 10 minutes is all about the AP test from last year, so just ignore that. I have, where did I put those videos? I put them in weird orders sometimes. You know what? I don't have an episode on nucleic acids or I don't have a thing on nucleic acids. You should definitely search one up. I'll see if I can find something cool. Maybe there is like a crash course chemistry or something, or crash course biology. That's always fun. I'll see if I can add more. Let's go to Teams though, and I'll show you some cool stuff on Teams. You know, I forgot to show you the lab from last video, so I'll make sure to do it this time. Okay. So if we go into files for unit 1, these are the notes, obviously. The notes on, I think under carbon, you'll find some useful notes. So that's notes that could be relevant. Assignments that are relevant. The maybe macromolecule overview, that might be good. None of this other stuff is very good. I don't have any labs either for this one. Yeah. So just some good stuff to know. Lots of things. I'll see if I can add more things that you might find useful. But there you go. We are going to talk about DNA so much. It is so important. It is at the heart of the central dogma, which is that whole idea that DNA is transcribed into RNA, which is translated into proteins. That is the central dogma. It's awesome. So enjoy that. And I will make another video about the other one, the other macromolecules. Hooray. All right. Toodles.